Hello young guys, good morning. You are welcome in soap biology classes. Today we will talk about biomolecules, the molecules which are found in living system. The main biomolecules are water, hydrogen monoxide, number 2 carbohydrates, number 3 proteins, 4 lipids and fats, 5th mineral salts, 6th nucleic acids, 7th vitamins, and number 8 hormones. So first of all, the carbohydrates. What are carbohydrates? In the beginning, in most of the books, it is described that they are hydrates of carbon. It doesn't mean that if you dissolve a carbon particle in water, it will become a carbohydrate. And generally it is said that they are compounds of carbon, hydrogen and oxygen in which the ratio between hydrogen and oxygen is 2:1. They are polymers of CH2ON. But you can see lactic acid is C3H6O3, but it is not a carbohydrate. Lactic acid is not a carbohydrate. The ratio between hydrogen and oxygen is same 2:1. On the other hand, deoxyribose sugar, 2-deoxyribose sugar, which is a part of DNA molecule, it is C5H10O4. So the ratio between hydrogen and oxygen is not 2:1, but it is a carbohydrate. So what is the exact correct definition of a carbohydrate? Actually, carbohydrates are polyhydroxyl aldehydes or ketones or their derivatives, acetals or ketones. They have many hydroxyl OH group and at least one aldehyde group CHO or a ketone group CO, but they are of many types: monosaccharide, disaccharide and polysaccharides. You know what is the smallest carbohydrate? No? Can you tell me what is the smallest carbohydrate? It is not described in most of the books. The smallest carbohydrate is glycoaldehyde, in which there is an aldehyde group CHO and one secondary alcoholic group CH2H. It is not found in living system, but in nature it is found. In the living system, the carbohydrates are monosaccharides, which are made up of one unit. They need not to be hydrolyzed while utilization in our body. Then there are disaccharides, which are made up of 2 monosaccharide units, and there are polysaccharides, which are made up of many units. The monosaccharides are of 2 types, one which has a ketones group CO, and some of them have aldehyde group CHO, so they are called aldones. One which have aldehyde group, they are called aldones. One which have a ketone group, they are called ketones. And there is a secondary alcoholic group always in the carbohydrate at the terminal end, where the carbon is attached with 2 hydrogen and 1 OH group. And in between all other carbon, generally there is 1 hydrogen group and 1 hydroxyl group. There may be difference in their isomerism, whether the hydrogen is present on this side and whether it is present on this side. It makes a difference in its isomerism. So first of all, the monosaccharides, they can be made up of 3 carbon, 4 carbon, 5 carbon or 6 carbon. The smallest from the 3 carbon group are dihydroxyacetone, in which there are 1 ketone group CO and the two carbon atoms, they are attached with two hydrogen atoms and two hydroxyl. So this is dihydroxyacetone or ketone. Its isomer, another 3 carbon compound is glyceraldehyde, in which we have one hydrogen attached with the carbon and a oxygen attached with the carbon. Here, if we may have two isomers, and this isomerism is compared from the terminal end, not from the end of functional group. Mind it. In most of the numbering is done counted by functional group. Here we have to count it from the secondary alcoholic group. So if the just adjacent carbon hydrogen is present on this side, then it is called D. If the hydrogen is present on this side, it is called L. So there are two isomers, D-glyceraldehyde and L-glyceraldehyde. So the monosaccharides may be made up of 3 carbon. They may be either glyceraldehyde or dihydroxyacetone. There may be 5 carbon monosaccharides also in our body, which are part of the nucleic acid, the ribose and deoxyribose. And the most popular one are fructose and glucose. All monosaccharides, many monosaccharides are C6H12O6. But what is the arrangement of hydrogen and hydroxyl group in the middle, that makes the isomeric binding. So in glucose, you have to remember only one thing. This is quite clear. Only one number we have in aldehyde group, and the 6 carbon have a secondary alcoholic group, 2 hydrogen and 1 OH. Right? Only variation is in the carbon number 2, 3, 4, 5. So the arrangement of glucose is 1, 2, 1, 1. Means 1, 2, 1, 1. And this will vary in other monosaccharides. For making a fructose molecule, simply this hydrogen and this hydrogen shifts to carbon number 1. It becomes attached with this one. This also becomes attached with this one. So here we develop CO, right? It becomes the fructose. Then we have disaccharides, which are made up of 2 monosaccharide units. They may be of 3 types: sucrose, maltose and lactose. Maltose is made up of 2 glucose molecules. Sucrose is made up of 1 glucose molecule plus 1 fructose molecule attached with each other by 1,4-alpha hydroxylic bond. In lactose or milk sugar, there is 1 glucose molecule, 1 galactose molecule. Here there is 1,4-beta linkage. Then just I will tell you what is this 1,4-alpha glycosidic bond and 1,4-beta glycosidic bond. Then we have polysaccharides. The polysaccharides are made up of many units. In plant state, there are 2 main polysaccharides, the cellulose and the second one is starch. Cellulose is made up of many glucose units which are attached with each other by 1,4-beta linkage. There may be starch, which is the storage form of carbohydrate in plants. It is made up of 2 types of molecules, amylose and amylopectin. In amylose, many glucose molecules are attached in a linear order with 1,4-alpha glycosidic linkage. In amylopectin, there is a branching. So at some places we have straight 1,4-alpha linkage, and at some places we have 1,6 linkage. In case of animals, the storage form of carbohydrate is glycogen. It is similar to amylopectin. Here also there is 1,4-alpha bond, 1,6-alpha linkage, but there is much more branching. One very important aspect of glucose is that the 4 valency bond of carbon, they are tetrahedrally arranged, and you know that angle in between two bonds is 109 degree, right? Not 90 degree. So what happens? Only 10% properties of a monosaccharide or disaccharide can be explained by a straight chain. But the molecule is not a straight chain. It is like this. One carbon is here, another carbon here. Then for the third carbon here, there is angle 109 degree. So here also 109 degree. So what happens? The hydrogen of the carbon number 4 and 5 come very close to oxygen of the carbon number 1, and they come together. So one way is to explain it by Fischer formula. Fischer formula is in which either the hydrogen of the carbon number 1 becomes attached with carbon number 1. So, but you know, this also doesn't explain the real structure of the molecule, because the real structure of molecule is something like this. So, Haber's projection was given later on, which is very much close to this structure. It is essential for understanding the bonding between disaccharide and a polysaccharide. So what happens? Actually, they are 6 carbon benzene ring-like structure, in which 1, 2, 3, 4, it becomes attached with the oxygen, attached with the carbon number 1, and the 6th carbon goes out of the ring. Here you can see that the carbon number 5 becomes attached with the carbon number 1, and the 6th carbon goes out of the ring. So this is called Haber's projection, in which the groups which are present on the left side, they are written on upper side, and with the groups which are on the right side, they are written just below. So you have simply to understand this thing. When 2 monosaccharide units, here carbon number 1, hydrogen and OH, here another monosaccharide in which carbon number 4, hydrogen and OH, right? And you know that what happens when 2 hydroxyl groups come very close to each other. Here also there is an OH, here also there is an OH. So one H2 molecule is eliminated and only one oxygen remains. So that oxygen is connecting carbon number 1 with carbon number 4. So it is called 1,4-alpha glycosidic linkage. So 2 things you have to remember. One, only 10% of the carbohydrates can be explained by straight chain formula. In 90% properties, they are cyclic benzene ring-like molecule. First of all, Fischer presented a formula, Fischer formula, and he said that, well, at the angle between different carbon atoms is 109 degree, 28 minutes. So hydrogen of the carbon number 4 or carbon number 5 come very close to oxygen attached with the carbon number 1. So either this hydrogen binds with it or this hydrogen binds with it. So it becomes a closed ring-like structure, like this. It is called Fischer formula. But it is also not very closely related with the exact molecular structure. So we have suggested another formula for its structure, in which the groups which are attached on the left side, they are written on outside, and the groups which are attached on the right side, they are written just below, right? And when two monosaccharides combine with each other, then here also there is one hydrogen, here there is an OH. So you know that whenever in between two molecules, two OH groups are there, water can be eliminated and only one oxygen atom remains. So it is called 1 carbon and 4 carbon, so it is 1,4-alpha glycosidic linkage, right? There is another possibility. In some of the disaccharides, what happens? The arrangement may be like this. Here hydrogen is just present just below, OH above. In this carbon, hydrogen is attached above, OH below. So this OH has to combine with this OH. So what will happen? This type of bond will be formed. So it is called 1,4-beta glycosidic linkage, right? So there is for breaking alpha glycosidic linkage, we need different type of enzyme. For breaking beta glycosidic linkage, we need different type of enzyme. Important thing is that in polysaccharides, we have, as I said you just now, that in maltose, 1,4-alpha glycosidic linkage is there. In polysaccharides, we have starch, glycogen, and cellulose. In cellulose found in plant cell wall, the many glucose molecules are attached with each other by this type of 1,4-beta glycosidic linkage, right? But most of all other bonding is 1,4-alpha glycosidic linkage. It is in case of starch, amylose and amylopectin. In amylose there is only one straight chain. In amylopectin we have one straight chain and there is branching with 1,6 linkage. In starch cellulose we have one beta glycoside. In glycogen also similar to amylopectin we have 1,4 and 1,6 alpha glycosidic linkage. But in glycogen there is much more branching. Thank you very much for today. Rest on the next one.